My name is Paola Neri. I'm a physician scientist at the University of Calgary. I'm a member of the hematology division at Tom Baker Cancer Center, but also a scientist, and I am a member of the Charbonnet Cancer Institute. I was invited today to discuss some of my research that was supported by your last 2015 grant. Translational research allows us to to see patient, get information from the patient, bring them to the lab, and use this information to discover more about the biology of a disease and with the idea to improve their outcome. And our group in the last 10 years has been focusing mainly on multiple myeloma, which is a kind of a blood cancer. Unfortunately, affect the plasma cells. The cells that are normally help us to fight the infection, to produce antibody, but when they become tumoral cancer cells, unfortunately, they start to grow very fast in the bone marrow, and unfortunately affect some of the organ in our body, especially the kidney and the bone. Unfortunately, it's an incurable disease, affect around 2,000 Canadians per year. Most of them, unfortunately, have a survival of only 3 to 5 years. And so the big challenge and the big question for us as a scientist is what we can do to improve their outcome. In the last years, there have been discovered and development on new drugs, but unfortunately, these drugs, they don't work for everybody. And some patients, even if they respond to some of them, they become resistant. And my research was mainly focused on, specifically focusing on a class of drugs called immunomodulatory drugs, IMiDs, and lenalidomide, thalidomide, pomalidomide, as you can see on this slide, are the main members of this family. What is unique about this drug, they have two mechanisms of action. One is a direct anti-tumor effect. They basically bind the cancer cells and they stop their growth and proliferation, affecting some important signal pathway for these cancer cells. But then they have also what we call now an immunomodulatory effect, meaning they are able to stimulate the immune system against the cancer. Even if they have improved the outcome of patients with myeloma, they don't work for all the patients. A third of them, unfortunately, develop resistance. And the focus of our research to understand why they become resistant. And through your support and the support from other groups, including our Myeloma Society, we established in our group a myeloma unit, or a myeloma genomic unit. As you can see in this slide, it's made of a big sequencer that allow us to really understand the genetic makeup of the cancer cells. And what this allow us to do in the last few years to understand more about the biology of this specific cancer, and more importantly, why these patients become resistant to certain kind of drug. And the final goal is, of course, to predict, to identify biomarkers. They can tell us what and why some patients become resistant, and it actually will tell us if we need to use that specific drug in the first place. Unfortunately, right now, we have a blind approach. We treat everybody in the same way, but unfortunately, later on, we discover some of them are sensitive, some of them are resistant. And so our goal will be eventually to have a biomarker, a way to predict in advance if that drug needs to be used for that patient. At the end of the day, we are trying to do what we call personalized therapy, so do the best treatment for that specific patient. So let me tell you a little bit more now about our finding, what we have discovered through your support. So we know that this protein called cereblon is essential for this class of drug to work, but we have seen taking sample from patients that, for example, some patients, they don't respond to this drug because they lose this protein. So the loss of this cereblon induces resistance to this Revlimid. But to make the story a bit more complicated, we have found that if you have cereblon on your tumor cells, unfortunately that doesn't mean automatically that they will be responding to the drug, because you may have what we call a special isoform, a special form of protein called isoform del-10 that unfortunately make you resistant, because in this protein there is really a missing part, as showing on this slide, that actually doesn't make the drug itself to bind to cereblon and then to act inside the cells. And this again is work done at the genomic level, using really interrogating the genomic part of our cells. As you can probably tell from my accent, I'm an Italian, and I did all my training there, my medical school and my fellowship, and around probably 6 or 7 years ago I moved to Calgary. I should say the passion for research was always in my mind. Unfortunately, I lost my dad when I was 14, and that probably triggered me the idea, and I wanted to do more about research, understand more about this kind of blood cancer. And I did my postdoc PhD in Boston in between 2003-2006, where I was very lucky to work with Dr. Anderson at Dana-Farber Cancer Institute, who is really a leader in the myeloma research.